The last thing I want to cover is this concept of mutation. So we're talking about, previous lecture we talked about replicating DNA and how there could be an error in that process. Now we're talking about transcription and translation. There can always be an error somewhere in that process. And so errors are going to basically result in what is called a mutation. A mutation is any change, let me get my pointer, any change in the nucleotide sequence of a cell's DNA. So what was is now a mutation, and it changes the DNA sequence. There are different types of mutations that can result. So first I want to talk about this idea of a substitution. So just like in previous images, my DNA is in blue, my RNA is in red. And here I can see the sequence of my DNA and its complement mRNA that's formed during transcription. And then I can see the different codons. AUG codon, that triplet, will code for the amino acid methionine. AAG for lysine, UUU, and so forth for each respective amino acid. In a substitution, you are substituting one particular amino acid, a substitution. So here I can actually see what was a G for the RNA. It substitute, instead of C and its complement being G, it incorrectly paired C with A during this step. That's incorrect. C's partner is G, but somehow that mRNA read an A during transcription. Well, GGC codes for glycine. AGC codes for serine. A substitution can change to a different amino acid. It could, it doesn't have to, but you're substituting the nucleotide. I'm going to talk about that in a bit. So one nucleotide changes in a substitution. Deletion and insertion are a little bit more deleterious in that they can create a lot of damage. We call that, so using our same example, we call that codons are triplets, AUG. It occurs in threes. If you take away a nucleotide completely, you don't even substitute it, you just take away an entire nucleotide, maybe they forgot to code for it, you are going to alter possibly not only that amino acid code sequence, but all subsequent ones, because now you've ruined the triplets. They're now no longer going to be read the same. So for instance, over here, UUU coded for PHE amino acid. Well, now it's no longer UUU, that U is taken out, it's UUG. Now it's leucine. Okay. Well, GGC was the next one for glycine. Now it's completely different. It's GCG because all those amino acids have, all those nucleotides have shifted because of that deletion. So that's quite deleterious for the protein. Insertion is a similar concept where it's quite deleterious, where you are affecting all subsequent codon readings. You are inserting an amino acid to where it didn't belong there. So you can have substitution, deletion, insertion. So these different types lead to different effects. So let's see what kinds of mutations we can have. A silent mutation is basically no change to the amino acid at all. Remember that multiple codons could account for the same amino acid. So if UUG, if we had a base substitution and that G switched to an A, well, UUA also coded for leucine. So that's called a silent mutation because there's no effect at all in that polypeptide. You still have the same amino acid resulting. A missense mutation, you're going to change the amino acid completely. So here I have UUG. It looks like that U changed to a C over here. So now UCG is going to code for serine. What was supposed to be a leucine? It's now coding for a completely different amino acid, a serine. That's called a missense mutation. A nonsense mutation is when an amino acid, what was an amino acid, it now changes to a stop codon in particular. So UUG, again, same example, coding for leucine. Now it's UAG, that U changes to an A. That's a stop codon, UAG. And now that protein will terminate at what was leucine, and that can be quite bad for the protein. Kind of depends where in the polypeptide chain that happens, but it is pretty much bad for the protein to have a stop codon there where it shouldn't have been there. The protein doesn't get its complete amino acid chain made. That's called a nonsense mutation. A frameshift mutation, I actually don't have an image for that one here. You guys have an image in the previous slide to where you saw nucleotide deletion and insertion. A frameshift mutation can be quite disastrous because you're shifting the codon readings. And so basically, you are affecting all subsequent amino acids that are going to be coded for after that frame shift. Maybe you're inserting a nucleotide, maybe you're deleting a nucleotide. That's going to have quite a disastrous result for that polypeptide. Basically, that protein is going to be non-functional if that happens.